I'm Paige.
I'm Hannah.
I'm Hannah.
I'm Allie.
I'm Mina.
And we did our project on genetic engineering.
Okay, so our abstract for our project. So due to newly introduced technologies such as CRISPR-Cas9, genetic engineering is a completely feasible possibility to alter the genome of organisms, primarily humans. So being able to directly manipulate the genetic makeup of humans is extremely controversial, as it defies the natural course of reproduction and raises many bioethical issues. In our project, we address the bioethicality of genetic engineering and its potential effects to the population in the future to come, such as reduced biodiversity. We created an art installation set in a futuristic dystopia in which all humans have been genetically engineered to possess similar qualities with an extremely reduced gene pool.
So before we start the project, we're going to show this video. Before we start the project, we're going to show this video that we made that explains CRISPR-Cas9, so you guys have some sort of background before we start.
It's not working. It's not working. It's not working.
Wait. Yeah. Okay. There. No. Yeah. It's good. It doesn't have sound for the beginning. So CRISPR-Cas9 is inspired by the natural defense mechanism of some bacteria. If a virus tries to attack a bacteria, the bacteria will destroy it by cutting up its DNA. The bacteria will save these pieces of DNA as mugshots in their own DNA, so they can spot a virus more quickly in the future. If a bacteria encounters a virus that they have seen before, they will enlist the help of Cas9, who will cut any DNA that matches the mugshot. When scientists wish to edit a gene, they create a complex with Cas9 and RNA. When the complex finds its way to the nucleus, the RNA will match to a specific block of code, like a mugshot. Then the complex will attach to a spacer, and the Cas9 will cut the DNA. Either the DNA will be destroyed, or scientists can insert a piece of DNA that they have programmed.
Okay, so that was our little introduction video to CRISPR-Cas9 and our concept/topic. So to clarify the topic, our group is focusing on human genetic engineering. So genetic engineering is the deliberate alteration of an organism's genome to express certain characteristics and produce favorable results that cater to the host environment. This technology has allowed for improved food quality, like GMOs, and increased profit for agricultural brands. However, for humans, it is extremely controversial because not many people have access to it, and also genetic modification would not only be used to cut out life-threatening disease-coding genes, but also to enhance people's physical features, making them more attractive. And it has the potential to greatly limit the gene pool and reduce biodiversity, which is essential to ensuring our survival in dire circumstances.
Decades ago, it was unimaginable to the human population to possess the ability to alter genetic makeup of organisms and control the expression of certain phenotypic genes. However, the discovery of recombinant DNA and in the 1970s and CRISPR-Cas9 in the past decade has allowed for humankind to imagine the impossible and manipulate the genome of an organism. With the development of such revolutionary technology also comes the bioethical issues humans must address, such as who can access this technology and to what extent humans can utilize genetic engineering on themselves.
So we drew inspiration from a lot of different resources. Just in general, CRISPR technology and other genetic engineering technology is written a lot about in science magazines, the media, and news. But in particular, what we used is the movie Gattaca, which we actually didn't know about before when we were generating this idea. And then we learned that Gattaca actually was very similar to our idea of like distinguishing between the two different social classes created by genetic engineering. So when we were able to watch that movie in this space, that really inspired us. In particular, that movie focused on the social aspect, in particular, like the financial opportunities of both classes. So that aspect really inspired us to focus on that. And then also something that we listened to was a CRISPR Radiolab podcast that talked about CRISPR technology and the presence of genetic engineering, and we thought that that was really interesting, and it made genetic engineering seem very tangible for the future, and that was something that really inspired us.
So now we're going to show some images and videos from our art installation that we did last night. So this is our intro video. So the whole art installation is like a simulation. So this is what you saw when you first walked into the installation.
You have chosen to advance your status from genetically unprivileged into genetically privileged. With reliable technology that has been used for years, your DNA sequence will be changed to make you better, more beautiful and healthier. The change in your status will greatly benefit you. If you choose to stay genetically unprivileged, you will be limited to designated jobs, schools, and living situations. In the advancement, the base pairs in your DNA will be directly changed, with no cutting or splicing involved. Proceed if you choose to continue with your advancement.
So as you can see, it's kind of set in a dystopian future.
So that was one video, and it was a 360, so that was one wall. And then this is a short animation that was displayed on another wall.
Yeah, that animation just showed what's that word?
Social class.
Yeah, the separation between the two social classes, between genetically superior organisms, humans, and genetically inferior organisms.
And then on the biggest wall, we had this playing.
And it's really just pictures of all the classmates edited to look like they've been genetically. The art was a big part of our project because that was the way that we displayed what it would look like. So it really took a lot of time, and that's like a big part of our installation.
Our goal was for the audience to look at themselves and like see themselves in this future dystopia.
And that particular wall demonstrated the loss of like genetic diversity because everyone started to look the same. So we had like each wall showing a different problem that could be associated with genetic engineering.
Okay, I think AK. Okay. Do you want to show it?
Yeah, go through the pictures.
Okay, so this is an insight into our simulation because we couldn't really have everyone go in there today.
Also, we all dressed up in a very futuristic costumes.
That's me and my friend Jimmy.
Okay, so our project proposal. So genetic engineering it has the potential to decrease biodiversity in humans. So when the gene pool is manipulated by humans themselves, there's a lower chance of survival in dire circumstances. So pretend there were to be like a huge disaster and it wipes out like half the population. Because the population is less diverse, there's a lower chance of survival. And genetic modification aids in eliminating disease-coding genes and expressing those that are favorable to one's environment. And although being able to select favorable traits in humankind may be useful for disease preventative measures, biodiversity is essential to the survival of the entire population.
Okay, so now we have a video about some of the pros and the cons of genetic modification. So one of the pros is that if we get genetic engineering right, we could really improve our quality of life, making us stronger and happier. That's what's showed in this picture. The second pro is that by genetically engineering animals, we could possibly make them more resistant to global warming. And for example, you could genetically engineer sea creatures to become more tolerant to rising ocean temperatures, and could it prevent extinction? The third pro is that genetic engineering could also allow us to bring back species that we have previously drove to extinction, and so that could do a big part in helping restore the environment. So now for the cons. So if we only genetically engineer those wealthy enough to afford it, we could create a new class system based on this, or further the class system that we already have. Another con is that, as Mina talked about, a lack of biodiversity is bad for a lot of reasons, but it could cause one disaster to wipe us all out. And then the last con is that because we don't 100% understand all the side effects of genetic modifications, it could cause some unintended consequences, so it's pretty risky.
So the impact of our installation was to spread awareness and have people think from the beginning and start to form their opinion on this, because it's in our future. And we want to illustrate both sides without bias and inform society, and to spark discussion and have people thinking about this now, and to explore advancements and possibly already think of solutions for when this becomes a problem, and recognize the possibilities and the negative consequences.
So for a conclusion, we hope that through our presentation and our art installation, viewers will be able to see the possible outcomes of genetic engineering, and they can form an opinion for themselves whether they think that genetic engineering should become the norm. While our installation was posed as a theoretical, we also wanted to show our viewers that this could happen not too far in the future. We also chose to do like a scientific side and an artistic side, because when we're viewing complex issues like that, you need a lot of different perspectives and a lot of interdisciplinary thinking to kind of figure it out. So which is what SciArt's all about.
Yeah, so like Paige said before, our ultimate goal was to generate discussion, because we weren't necessarily trying to like find a concrete solution, because the problem is so new and will be very impactful for like future generations. We kind of just wanted to get the discussion started. So while most people will agree that genetic engineering should be used to like cure disease and all of that stuff, at what point does it become too much? More people will disagree that genetic engineering should be used to make people more intelligent, and then even more will disagree that it should be made to make people even more attractive. So it's up to you to decide what exactly you value and how you think that this technology should be used.
And here's just some of our works cited. Thank you.
Imagine the impossible.